Good afternoon, everyone. I'm John Thompson, the Catskill Regional Invasive Species Partnership Director. And today we're starting one of our Catskill Regional Invasive Species Partnership virtual meetings. Well, we have a great meeting today with some great presentations lined up, including two presentations on rapidly spreading invasive species in the CRISP region. Before we get too far along, I'll ask each of you to introduce yourselves in the chat so that we have an idea of who's here today. And just a reminder to please mute yourselves as feedback and background noise can interfere with others who are speaking. I could also ask you to do one more thing, and that would be we're lining up a potential partners meeting that would be in person in the end of May. If somebody has ideas about where to hold that meeting, we try to rotate the in-person partner meetings around the region. Then that would be great. I could look into it. You could always email me about locations to have meetings and or give me a call. So before we get too far along, wanted to just remind everyone that closed captioning is available at the bottom of your screen. You can hover over the dots at the bottom of your screen and click more. You should see CC for closed captioning. On the screen in front of you, you should be able to see the agenda for today's meeting. We switched up the agenda a little bit. We have a substitute speaker, Jessica Cancelleri, is going to give our first presentation today. And we'll start off the meeting with her presentation and Liam's after that. I'll give some highlights from CRISP. Then we wanted to acknowledge the project that we selected for the 2024 CRISP RFP. We'll also acknowledge some of the new signers of the CRISP cooperation agreement. There'll be some time for partner updates after that if people have announcements or updates that they'd like to mention. So I'd like to quickly move on to our presentation, and I can stop sharing, and I'll introduce Jessica Cancelleri. She is Forest Health Section Chief at DEC's Bureau of Invasive Species and Ecosystem Health. She's a research scientist who formerly managed the Forest Health Diagnostic Lab and took over the Section Chief position last June, which oversees the lab and the rest of the Forest Health program. I'd like to thank Jessica. She was able to step in the last minute when Kelsey had to bow out. She's had some traveling difficulties, so she wasn't able to get back today. But you can go ahead, Jessica, when you're ready. Thank you.
All right. Thanks, John. Thanks, everybody. Yeah, I'm going to focus today on beach leaf disease research updates. I know that Kelsey, who was supposed to present today, had presented, I think, to the same group maybe in November and had given some survey updates. So she asked me to focus a little bit more on research. And that being said, I'm going to give you, there's a lot going on. So I'm going to give kind of a general rundown of things. Some of these topics I'm not an expert on. And so you can contact me afterwards and I can put you in touch with the researcher or the publications and such that I'm referring to if you want more information. But I'm going to start by giving just a quick overview again of beach leaf disease. And I think most of you have probably seen these maps and stuff at some point in time, but I think a quick review is warranted. So beach leaf disease was found in New York in 2018, although we did find some Forest Service records that indicated it might have been here as early as 2014. And that's why Cattaraugus County there in the west is in blue on this map. But the regional spread has been extremely rapid, and it's really ramped up in recent years. And you can see kind of the exponential rise in total counties infected on the right. That's a map representing this regional spread. You can see that it just really ramped up starting, around 2020 or so. And just to think a disease like beach bark disease took 60 years just to spread throughout the Hudson Valley. And you look at what's happened just since about 2018 or so. It was pretty restricted to Ohio for a while. And then since it hit Western and Eastern New York, it's kind of just gone crazy. So that's the current regional status. It's been found in Ontario. as well as states as far south as Virginia and West Virginia, and all the way up into northern Maine. And here in New York, this is our current map, as late as July. So again, I think Kelsey presented this stuff to you, and obviously our survey results haven't changed since. But it just kind of shows that, Long Island and the lower Hudson Valley and Western New York are the most severely impacted so far, and where we have the most counties infected. These were also the first places it showed up. Again, Western New York, sometime around 2018. Long Island, the very next year, I believe we found it, 2019. And now it's spreading across the state pretty rapidly. We found it in the Adirondacks last year, and we're anticipating several more counties up in northern New York this year. And one of our staff, Maria Moscali, did a spread study out in Portland County, New York. And I like to show people this because it just shows kind of the pattern on the landscape within a single stand of how the disease spreads. So this first figure shows expansion by year starting in 2020. And we started with a 1 acre spot, with several trees infested. By 2021, it had grown to about 5 acres. Then in 2022, almost 16 acres. In addition to the spread, from the inside out, we would also see all these satellite infestations, which are represented in these blue dots here. So those start as a few single trees, and then they start spreading out just like the one that here in the center that we've been monitoring. So it gets really crazy around 2023, when suddenly the 15 acres spreads to almost 500 acres. And I'm sure it's even bigger than this, but we started running into trouble surveying with just private property and such. And we're going to work with regional foresters this year and some private landowners to try to add to this map. But we kind of just stopped monitoring because we just couldn't go any further. And this is in a hardwood forest. It might be like an Allegheny hardwood forest. So beech is pretty prevalent, but there's also other hardwood species present as well. In New York, we've been monitoring beech leaf disease since 2018 at several monitoring plots. We have, I can't remember the total, I think 25 or 26 in New York, but as part of a larger network across the entire Northeast and into the Midwest, with about 150 plots total. And I'm not going to present on any specific results from that today, but I just wanted to make you aware that this was going on. And these plots measure, they're 10th acre plots, and they measure things like canopy health and also regeneration, forest structure, mortality. So we're starting to get some pretty good long-term data that's telling us a little bit about how it spreads within a stand, how it impacts trees, how things like beech bark disease interact with beech leaf disease, and so on. So that'll be pretty interesting. It's such a huge data set as well. So to get more into the research updates, when we first started looking at this disease, it was brand new. As many of you probably know, it's associated with a nematode. And I think the jury is still out on whether the nematode is actually exotic or not. It's not quite related to, there is a closely related nematode in Japan, but the one in North America has been labeled as a subspecies of that one, and it does not infect Japanese beech. And so it's thought that it is a separate subspecies, and it could be native. We just don't know. But for a long time, for the first several years of tracking the disease, there really wasn't much known about the physiological impacts or the cellular impacts of beech leaf disease. And some of those studies have just come out like in the last couple of years. So one of those was published by USDA, ARS, and it looked at the cellular impacts of beech leaf disease. And it showed, as you see the symptoms in the leaf, you see a lot of leaf banding and leaf thickening and textural changes. And so when they looked closer at that, they found that, it's exactly what it looks like. The leaves that are infected with the nematode are larger in size, they're thicker, they're denser, they have more chloroplasts, bigger nuclei, and they're extremely unorganized. So I'll show you a picture of what that looks like with an electron scanning microscope. So the photo on the left shows healthy leaf tissue, and the photo on the right, of course, shows the tissue with the nematodes. And it's interesting, you can just see that disorganized structure. It kind of just takes all these layers of mesophyll and makes them just all mixed together. It kind of reminds me of like what jumping worms in a way do to the soil, how they homogenize all these distinct layers. It's something kind of similar to that. And it's not exactly visible in this photo, but there is an actual leaf thickening that occurs. So like in photo B, that layer of leaf tissue is now actually wider and denser than leaf tissue A. And so, let me just see here. So as these studies have been done, like one thing they've been looking at specifically are things like leaf veins and stomates, and how does BLD affect those structures? Lila Fletcher, who is with the Yale School of Environment, or I think that's what it's called, the Yale School of the Environment. Her and her team have been working on looking at some of these features. And one thing they found was that symptomatic leaves had wider vein structure, not so much the main veins, but the really skinny minor veins end up being wider and actually less dense. And so, whereas like the leaf tissue as a whole becomes denser, the veins actually become less dense. And so at least in the short term, they found that the leaf is still conducting just as much water. There's fewer like cells in the veins, so it's not able to hold as much water, but because the veins are wider, it's actually able to move more water through it. So that was actually asymptomatic and symptomatic leaves were equal in that regard. So water conductance hasn't been impacted that much. So that's good. And then, however, though, they looked at, I'm just trying to see, my screen is being blocked by my face. Let me just move this. That's actually Lila up there in the photo. And this was from a research meeting. And if you look at the photo over on the right of, that's like that circular structure coming out of the vein is actually, that's a vein coming out. I can't see it with my thing, but the round piece there. And what it's showing is the difference between healthy tissue on the right and unhealthy tissue on the left. So that really thick piece of the leaf tissue on the left is infected. So when I talked earlier about this like increased density of leaf tissue, you can kind of see it in that picture. And so because they're thicker, they take more resources to grow. It's just like more tissue to support and to build. And so because of that, although they're using more resources to build that tissue, they're producing fewer sugars. And I'll talk about why in a second. And so all of this, again, like nets in this sort of compromised carbon budget. You know, carbon is essential for plants to grow. And without it, you know, they're going to be in like this long-term deficit, which eventually leads to things like, you know, long-term decline and dieback, low vigor, increased stress, and decreased tolerance of, you know, environmental stressors. So the reason that their photosynthesis is lower is because, and I just want to look, I'm sorry to stop I was hoping to have this slide before these so I could explain. So I'm just going to skip here and then go back. So some more physiological research that came out just last year was from Cameron McIntyre at the Forest Service. And he was really the first one to look at some of the physiological impacts of BLD. And his paper is really intense and dense and full of a lot of information. So I'm just going to briefly summarize it, that he found that infected leaves did have reduced photosynthetic capacity, reduced carbon assimilation, reduced water efficiency, and increased leaf nitrogen, which might sound like a good thing, but all in all, the increase in leaf nitrogen wasn't enough of a benefit to offset all the other negative impacts. So again, we have this compromised carbon budget. And so he did a lot looking at water efficiency and gas leaf exchange. So when it comes to the gas exchange, you know, leaves, they absorb carbon dioxide from the environment through their stomata. And he found that there were issues there that, and other researchers have also found that the stomatal openings get distorted when they're infected with BLD. And because they're not able to absorb as much CO2, they have reduced photosynthetic capacity. And also they're losing more water because the stomatal openings don't maybe open and close as they should. And when they open, they're more prone to losing water and moisture from the leaf. So going back to this photo, Lynn Carter got a photograph of healthy stomates versus diseased ones. And you can see that on the right, they're just like all distorted and you can't even really make out the openings. Like here and there you can, like down in the lower left corner, I think those are some openings there. But as you can see, like gas exchange is obviously going to be impacted. And so as stomatal conductance decreases, photosynthetic capacity decreases. So it's all these little things interacting just to, in general, reduce tree vigor. But it's interesting, and because this is all very new information for us. There's also some research going on right here in New York State. And before I go into the slide, I want to say that these two gentlemen, Justin and Andy, presented for us just this past week at our beach leaf disease communications meeting. And John, I'll send some information to you. If anyone on this call wants to be part of that, we have a listserv and we send announcements when we're going to have meetings and presentations, and I can put you on that. I also have the recording of this specific presentation. If you'd like to hear it, I'm happy to share it with anybody. But they set up 35 monitoring plots along a urban-rural gradient from New York City all the way to the Catskills. And they're in the middle of this project, like year 2 of 3 or 4. So they don't have all their results yet, but they're essentially looking at understory impacts of beach leaf disease, looking at site-specific impacts to see, is the disease more impactful in suburban areas, urban areas? They're looking at invasive colonization. So some of the take-homes from their presentation this past week were that suburban beach are more heavily impacted than urban beach. And they didn't really go into why, but I think it just has something to do with the things that we think spread beach leaf disease, which is, predominantly probably birds and wind and rain. So obviously with birds, there's probably just more birds present in suburban areas than there are in the middle of New York City. They also found that the disease appeared to be progressing faster here in New York than it is in the Midwest. And some of you may have heard these figures. I think out of Ohio, they're saying saplings can die in 2 to 3 years and mature trees can die in 7 to 10 years. And based on their preliminary data, they think that's happening faster, which was a surprise to me because in our monitoring plots, we haven't seen that much mortality yet. When we do see mortality, it's hard to definitively blame it on beach leaf disease. So I was more optimistic and thinking that it might be slower here, but apparently that's not the case. And one reason they think for that is the interaction with beach bark disease. In Ohio, they, I don't know, either don't have it or they don't have as much of it. They're more on the front of that expansion. And so in those places where we have higher levels of beach bark disease here in New York, they do appear to be interacting and impacting each other. And then he also showed this really interesting photo of Japanese stiltgrass invasion after a beach leaf disease infestation. As you know, the disease results in canopy thinning, increased light getting into the understory. And our partners, down in Westchester County early on, this photo is from Westchester County, recognized that this was going to happen. And many of them have already done what they can to implement, mitigation efforts for controlling invasive spread. But, we've talked about it a long time, but I thought it was really impactful actually to see this photograph. It's something we assumed was going to happen, but we didn't have hard data on it. And so it's extremely interesting. And the scary thing is, it's just like the timeline on this. Beech trees are extremely shade tolerant and they provide a lot of shade in the forest, and they're very long-lived, later successional species. And so, and getting increased light into the forest, that's something that's going to persist. for decades and can't easily be replaced just by planting, new young trees. So it's something that land managers are going to have to cope with in the long term. Let me just make sure. So some other research that's going on is looking at the microbiome of beach and what other organisms may be involved. Because I think researchers are looking at nematodes and how they've behaved in the past. And then they're looking at the way beach leaf disease is behaving. And they kind of assume there might be other things at play because nematodes do cause damage, but they don't usually move as fast and cause the level of damage that we've seen with beach leaf disease. So some preliminary work was done to look at bacteria specifically that were found on only symptomatic leaves. And so the 4 bacteria on the left were 4 that were identified as only being present in symptomatic leaves. And some of them do have historical associations with insects and plants as sort of either coexisting organisms or harmful organisms. But I think at this point, not much else has been established other than this is a possibility, but we don't know specifically how any of these bacterium are interacting with the nematode. I think I have a slide on biocontrol later. I can talk about people that are using bacteria for potential biocontrol as well. And then there's a whole bunch of genomic research going on, and this is by no means captures all of it. It's just kind of like a sampling of some of the stuff that's happening with some of our regional partners. And a lot of it is focused on just population dynamics, like using DNA to understand what the population here consists of. Is it one population? Is it many? How genetically diverse is it? Are these separate introductions? If it is non-native, are these like separate introductions into New York State and the region, or are they all separate? Because I think it'll tell a lot about the potential impact. If there's a lot of genetic diversity, it might be harder to control on some level because you'll have all these sort of localized populations with different genetic modifications. And if it's one population, that could potentially be advantageous for future control efforts. And again, this isn't really like my wheelhouse. I can't talk about genomic studies in any great detail, but if you want more information about any of these, I'm happy to share the papers. At ESF, we're working with a forest pathologist there, Shannon Lynch, and she's been trying to start a project where she wanted to identify the entire microbiome of beach in general, and then find ways to like modify that microbiome to confer greater resistance to the tree. And I like to explain it as like probiotics for trees. And that work is really, really early on. And it's part of a larger effort at ESF. It's kind of integrated into their tree restoration program, where they're building these foundational concepts to like apply to other tree species eventually as well. And then, of course, at DEC, we are trying, currently there are no forest treatments, right, for beach leaf disease. It's all restricted to individual tree treatments in more of a landscape setting. So we're trying to develop treatments in the forest. And as part of that, we're partnering with the Forest Service and ESF to do silviculture for beach. And those treatments, we're actually marking trees this month, and those treatments will be done this fall and winter. And it involves 2 separate treatments, an overstory canopy thinning to increase air circulation and light into the stand, which we think might desiccate the nematode and reduce its capacity to proliferate. And then the other treatment is understory beach removal, where we're removing understory beach in the hopes that it will break up the vertical ladder of inoculum that we suspect, is the way that nematodes are moving through the canopy. And then, of course, control. So absolutely no data on this yet, but it's coming soon. And as of now, the 2 most effective treatments that have been shown for beach leaf disease are phosphites and nematicides. Polyphosphate 30 is a potassium-based fertilizer product. It's not an insecticide. And it works by increasing tree vigor more than it actually kills the nematodes. Although it also like decreases cell division. And because we know that when a beech leaf is infected with beech leaf disease, it gets, remember, like wider and more dense and more cells and more everything. So it does prohibit that cell division a little bit. And that's applied as a soil application, ideally twice a year. Most practitioners I know are doing it in like June and July, or July and August, or whenever they can during the growing season. And currently, SUNY ESF is in the process of trying to get funding to take the phosphite work and apply it to a forest scale. I can't recall if I have a slide on that after, but they're also testing a cheaper potassium chloride product that would make it a little bit more financially feasible for people to use these potassium-based products. And Broadform is currently the most effective pesticide that's been labeled for beech leaf disease. There's just some uncertainty about timing because you can apply it early in the season when the leaves are tender and they're more likely to take up the product, but it also probably doesn't kill the eggs. And so if the product can stay in the leaf though, until the life stages hatch, it would be toxic to them. The other thought is just to treat sometime during the summer before the nematodes move into the buds, which we think happens, towards like late summer to fall is when they start moving into the buds. And so if you can prevent that from happening, that would be effective in reducing next year's population. Oh yeah, so here it is. This is ESF's proposed research. So I already described that. And on the right is just a picture of how these phosphite products are applied. There's a trench that's dug around the tree, and it's not usually blue like that. URI just did that so you could see it. And so they also plan, if they get the funding, their focus is also on like proper dosage and also evaluating non-target impacts, which is really cool because we know some landowners and managers that have tried phosphates in their forest, and they would come to us with questions like, I'm trying this, but I have no idea of the dosage to use. The label doesn't cover an application like this, and we didn't know either. It's just like one big experiment. So the fact that there's a group out there trying to optimize dosage and economic efficiency of these treatments is pretty cool. And there's also a proposal in for the very foundation of biocontrol work starting at Pennsylvania State University. But Dr. Cantor here is working with a whole lot of partners across the region to, it looks like they've already identified some potential native bacterial biocontrols for BLD. And so now the next step is to isolate them and test their nematocidal properties, in Petri dishes, essentially. And then also take those bacteria and test them in greenhouse settings on actual plants to see if they're killing beneficial organisms. And this is just a slide that I always have at the end of BLD presentations showing some of the take-home messages about the disease and what we know about it right now. I guess, what has changed since I last made this was that data from New York that came out saying that mortality might be happening sooner than we thought. My third bullet there of slow decline, it might not end up being as slow as we think. In New York, we've had some drought. We had a drought the last year or the year before, and we saw some really increased severity levels across the region. And I think a lot of it had to do with that. Beech, does not tolerate drought very well. So I think environmental factors are going to play a big role in annual symptom severity. But the other thing I'll say is that we've seen high variation in symptom severity. So if it's a good year and environmental factors, are more supportive of beech growing conditions, then we do see reduced severity levels. So that kind of says that trees might have the opportunity to sort of catch up and kind of maintain in between years, and then be a little bit better prepared for when a more severe year comes. But I think we really just don't know yet. And that's all. Sorry, I went a little over.
That's great. Thank you, Jess. That's an excellent presentation. And it's really interesting to see some of the treatments that are being worked on happening really quickly for something that we've well, you've learned a lot about over the last couple of years.
Yeah, there's a
So,
lot going on. It's hard to keep up with all the latest research now, and it's a lot of stuff happening.
yeah, that's good. I know that you need to leave pretty soon, but if anyone has any questions for Jess before she takes off, you could unmute yourself and ask right away, or if you wanted to enter them in the chat. I don't see any questions yet.
Yeah, and just feel free to email me too if there's something specific. Like I said, we have a lot of resources that we can put you in touch with, and if you want to be on our, the point of those BLD meetings is to inform all of our New York State partners of the latest research and happenings. So I'm happy to put you on that list. I'll send maybe John the information, and then you can send it to your partners. But that's kind of the best way we have right now to stay up to date on everything happening in New York. They don't happen every month. They're kind of like every few months right now.
Great. Jack, I see your hand raised. Go ahead.
I just presumably these treatments that groups are working on would be for individual trees or for small stands. Do we have any thoughts about an attempt to address the issue at the landscape scale?
Well, I'd say at the landscape scale, the efforts are more focused on like genetic conservation right now, not knowing how it's going to play out, like increased seed collection, and then tying in with kind of like the microbiome work I talked about earlier, just kind of looking at it from a different perspective, or at least trying to conserve beach genetics for now, just in case it all goes really badly. But no, the only other like landscape level treatments I would say would be biocontrol, right? But that's going to take many, many years, and they're just starting to approach that now. And even like the silviculture, that was aimed at a forested landscape, but it's not something that could easily be implemented by many land managers. So, even like in our own forester community, beech is not a timber species. They generally don't manage for beech. So, how many people even are we going to get that are going to be willing to execute, beech silviculture work on their property? So no, not really. But the closest thing I've seen is like the silviculture and the phosphite treatments at a forested scale, but that's still going to be limited to someone's, I'm guessing 20 or 30 or 100-acre stand. But yeah, that's all I got for now, unfortunately. And so our goal, early on in our program management, knowing that there were no treatments available, we started trying to think ahead and what do we need to do to preserve genetics? So that's a question that we're still kind of answering. It just moves slowly.
Thank you.
Mm-hmm.
Jess, I know you got to get going.
Thanks,
You're
everybody.
welcome to hang out if you can.
No, it's all right. I'm going to go. Thanks. Nice to see you.
Thank you so much for stepping in today. I really appreciate the presentation. That's a lot of great information to share with us. I really
Anytime.
appreciate it.
Yes, my pleasure. Thanks.
Thank you. So next we'll move on to Liam Summers, and you can go ahead and share your slides, Liam. Liam has worked as the entomologist for the DEC's Forest Health Diagnostic Lab for over 5 years, providing tree pest and insect identification services to the public, as well as processing samples from the DEC's various invasive species trapping projects. Thank you for joining us today, Liam. We really look forward to your presentation.
Absolutely, yeah. Thank you for having me here. So like John mentioned, I am an entomologist here at the Forest Health Diagnostic Lab, and today I'll be talking about elm zigzag sawfly and some of the history in New York State. So it is an exotic pest native to Eastern Asia. It's found in some parts of China and Japan, and it's been spreading through Europe since 2003. It's been found in 19 European countries, I believe. It's probably over that number at this point. I believe that was a few years old. But they found it to have 4 to 6 generations a year, so it does reproduce very quickly. And the first detection in North America was in 2020 in Quebec, Canada, from an iNaturalist submission that I've actually have clipped over here on the right-hand side. You can see that it does make a pretty distinctive zigzag pattern, hence the name. It's named very appropriately. Usually it will make that sinuating pattern between the leaf veins there. It usually stays in between them. Sometimes I'll go over, but generally it's in that spot. And there's nothing else that makes a pattern quite like this in elm leaves. So it is pretty diagnostic of this species. Since that Canadian detection in 2020, it's been found throughout much of the East Coast, all the way south to North Carolina and Vermont, Ohio, New Jersey, and Massachusetts just being added to this list pretty late last year. We do think it's been here for a little bit earlier than that initial detection. We found some pretty suspicious bug guide submissions from 2018 that looked a lot like the adults. But you know, that's kind of how it goes in a lot of these cases. People don't really know exactly what they're looking at, and it just takes that information being put in front of an expert's face to first realize what it is, and then, you know, the alert goes out to everybody, then everybody looks for it, and then all of a sudden you just find it all over the place. So that's what seems to be happening here. Now it is a defoliator with the potential to cause severe defoliation damage. These photos here on the right-hand side were from the very first detection in North Carolina. This was on somebody's backyard ornamental elm tree. I believe this was an American elm. They were wondering what was going on, and I believe they called the state of North Carolina, and somebody came out there and recognized it as elm zigzag sawfly. You can see that tree there is almost entirely defoliated up to the top of that crown. That's what they'll normally do. They'll start from the bottom and just start working their way up to the top there. And that photo on the right is after they treated it with an insecticide. We honestly didn't even think that photo was real at first because we couldn't believe how many of the larvae there were on the ground. But they were, I mean, they were everywhere. And one of the main concerns in Europe is that it's outcompeting some of their native species that rely on elm as a main food source. I believe there's a moth and a butterfly species that are both threatened in Europe that elm zigzag sawfly is constantly outcompeting, further exacerbating that species. And this is just another layer of stress added on to an already declining elm genus, right? Because we've had Dutch elm disease that has wiped out millions of elm across North America. This is just, you know, another layer of bad news for elm, essentially. So some of the identification and life cycle, starting out with the eggs, they are extremely small. As you can see in this photo, they almost look like a drop of rain on the edge of the serrated leaf. It's just right on the tip there. They're usually under 1 millimeter long and wide. I can't expect anybody to find one of these, but in severe cases, they can lay up to 49 eggs on one leaf. And they'll usually be in this life stage for about a week. And the larva, which is the most commonly encountered life stage because they are the most longest lived and easily identifiable. You know, they range in quite a bit of sizes because there's 6 larval instars starting at under 2 millimeters long and growing up to about 11. The main identifying features for this is the black band across the eye and the black capital T-shaped markings on the second and third pair of legs. In the bottom photo there, you can see the little red arrows pointing to those features. It's not always as clear as it is in that photo. You know, like I said, when they're really small, it's tough to pinpoint those features. So it's something that you would need like a hand lens or have to bring it back to a lab to use a microscope to really see these features clearly. But they will have both of these features present. Because there are other sawfly larvae that look a lot like this, but they don't have both the black band across the eye and the black capital T shaped markings. And next is the pupa. It kind of looks like a loosely woven meshy cocoon, usually on the underside of the leaf that they were previously feeding on. They'll be in this stage for about a week. They do also make an overwintering cocoon. This is pretty new to some of our phenology research. I should have added a photo in here too, because it looks a little different. It's more of like it looks more like burlap material. It's not as see-through as these that I have photographed here. But those will usually be down in the leaf litter, and they're very inconspicuous. You know, you can imagine like a very small brown oval, you know, really just mixed in with a bunch of leaf litter and organic matter. It's tough to really find that. But during the growing season, this is what you'll see if you see a pupa. And the adult, pretty non-conspicuous. It looks a lot just like a black fly. It isn't really a fly, even though it has fly in the name. It is a hymenopteran, so they are related to wasps, bees, and ants. But they're almost entirely black, except for the yellowish pale legs. And they're pretty small too. They're 6 to 7 millimeters long. They're very quick moving, so they're tough to see out in the wild. It's something that you kind of have to catch in a trap. And they're known to be great flyers as well. You know, even for such a small insect, they can fly up to 56 miles a year, which is unbelievable. Not many insects can really do that, especially at that size. But they're pretty short-lived, only living 1 to 6 days in this adult stage. So this is typically what you'll see in an infested area. Generally, what we found in New York is more like photos on the far left there, where it'll only be like a feeding pattern or two on like a few leaves throughout like a single tree. You know, basically all the feeding level that we found in New York State has been pretty low and is just looks a lot like that left-hand side, but it can, in certain situations, eat its way all the way down to the midrib of the leaf, like on the far right-hand side there. They kind of eat all the leaf tissue, kind of not leaving behind that diagnostic zigzag pattern. So it can be a little tricky if it gets to that point, but oftentimes if you're seeing leaves, you know, fed down to that level, you'll be able to find a specimen to correctly identify it as EZS. So here's what the elm looks like in New York State. As you can see, there's not really a whole lot there, which you can thank Dutch elm disease for. But right along that St. Lawrence Seaway area in St. Lawrence and Jefferson County, it has the highest concentration of elm, which is where we focused a lot of our surveying efforts in 2020, I believe it was late of the year 2020, right after Canada found their first EZS. They contacted us telling us to just keep an eye out for it. So then the next year in 2021, we set up some traps along that seaway area, which we didn't find anything. Kind of a crapshoot. You know, we went to like 3 or 4 different sites to just set up the yellow sticky cards and some elm trees. You know, just kind of seeing if there's any chance we could find it. And, you know, not to our surprise, we didn't find it. So then the following year, we did some visual surveying up in that area, and we found it at 3 state-owned sites at Wilson Hill, WMA, Lost Nation State Forest, and Brasher State Forest. And you can see they're kind of all in a little cluster there, right at the top of St. Lawrence County. So, you know, after finding it in those 3 areas, we did a lot of surveying around. We couldn't find it anywhere else except for that one area. So we're kind of thinking like maybe it was secluded in this one spot. Wasn't far from the Canadian border, not too far from Ontario, where there was a boatload of our naturalist submissions positive, I should say. So there was a bunch of EZS all throughout that area. So we're kind of thinking maybe it had just made its way into New York State. Also, like I mentioned, the defoliation damage was very minimal. And out of all the trees we surveyed, we estimated it was about under 10% of the trees had at least one zigzag feeding pattern on them. So still very low levels, very sporadically in these areas that we found it. And that was our thought up until June of last year, when a homeowner contacted a DEC forester saying, hey, like all the leaves on my elm trees in my backyards are being eaten by something. He went there, took a bunch of photos and sent it to our lab, and we couldn't believe it. That is big elm zigzag sawfly 150 miles south of that original detection. And this is still the highest level of defoliation we've seen to date. You can see those photos in the back side. Most of those are elm, and you can see, I mean, a lot of those trees are pretty see-through there because all the leaves are really gone. There's all life stages present there. They seem to be happy living there, just going from tree to tree, not having to move too far. So that was a very favorable ecosystem for them there. After that detection, we contacted our outreach team. They put out some social media posts encouraging the public and foresters to keep an eye out for it and report it through IMAP or send it to our lab. And pretty soon after, we filled out this map, finding it across almost the entire state of New York. You know, it's just, we weren't really expecting any of that, but you know, it's just one of those things, like you put out a little bit of outreach and then all of a sudden you find it everywhere. So this is what our map looks like now. It's still all the same counties, just with Washington County added to the previous map there. So you can see we have some pretty good coverage across the state. And fortunately, we have such a great tool like IMAP invasives that we're able to basically survey the entire state of New York without really having to go too far, because folks can enter areas that they surveyed and found it or not found it, which makes our job so much easier because we only have 4 staff members here. So, you know, you can see that it just takes up a good portion of the state there. And one of the main areas of concern in terms of damage severity is with urban elm. That is one of the areas that has the highest reported damage in Europe, is in some of the urban settings. We've used in North America, we use elm as a common street tree. There's so many streets named Elm Street, right? And that's generally because arborists would use elm to line up and down streets because it was a very low maintenance tree. They're very gorgeous. They grow tall and straight. And this is a great environment for EZS because, you know, like I said, on the Niskayuna detection, they don't really have to go far. You know, they're able to just move tree to tree to tree, keep reproducing until, you know, they've basically run themselves out of resource. Along with street trees, there's also ornamental trees like the North Carolina detection. I reposted that image because that is an ornamental tree just planted in somebody's backyard. Oftentimes in these cases, they're able to grow nice and big, you know, without much competition. So they just have a wide open area to get into a huge mature tree. And again, they don't have to move far. They're able to keep reproducing in that same area and just move themselves all the way throughout the resource there. Most of the time, management isn't necessary, especially in New York State, where we really haven't found it to have high levels of damage. But fortunately, there are like a number of insecticides that are effective against EZS. You know, the larval stage is pretty easy to control because they're such a soft-bodied insect, right? So they're pretty vulnerable to insecticides. Things like horticultural oils work pretty well, but then there's also sawfly specific insecticides like spinosad, which is shown to be very effective. And Virginia has been doing a lot of research on this front. They've been doing some research with dinotefuran showing that it's been very successful as a preventative and just a control measure. But, you know, again, fortunately, we haven't really had to deal with this yet in New York. And we're pretty lucky because there's Massachusetts just recently found like a whole area that was pretty defoliated from EZS. So they seem to have a lot higher damage than us. Same with some of the southern states like North Carolina and Virginia. They have seemed to have been getting it a lot worse than us up here. So some of the research we conducted last year, partnering with SUNY ESF and Melissa Furkey, who's the entomology professor there, she's been helping us conduct a lot of this research. We were just trying to pin down how many generations a year. It looks to be around 2 to 4 here in New York. It is an asexually reproducing insect as well, which just kind of aids to the quick life cycle and turnover, and which also aids the spread if you're just constantly pumping out new generations. We're also trying to pin down what native predators, pathogens, and associate insect community that might be controlling this population looks like. Melissa did take all these great photos here on the right-hand side. She did find there are some pathogens that seem to be controlling some of the larval stages. There's been plenty of times where we went out to a positively detected area and we found, you know, a few leaves with some of that feeding pattern, but there's just no insect there or specimen to back that up. So something is feeding on it, something is controlling these populations. So we tried, you know, doing some research on that last year. We'll be continuing that research again this year. We did a trap comparison study using some different funnel traps and a couple other different methods, which the yellow sticky card came to be the best method for collecting the adults. And the funnel traps were pretty non-effective at collecting that. But this is generally just like a detection method. We're kind of working out like, you know, ways to use the number of adults on a sticky card to predict how big the population is in that area. And we have a multi-state working group that we continually have calls with. I think it's like every few months just to collaborate and see what the damage is looking like across the East Coast. And all those other states that I have on one of the previous slides are usually in attendance there. And it's just kind of a big collaborative effort to just, you know, see the differences across state to state, what EZS really looks like and how bad the damage is. So this is what some of our yellow sticky card trapping results looked like. You know, like I said, we found it to have about 2 to 4 generations a year here. You know, it's one of those things, like a lot of insects, the more south you go, the more generations you have. But, you know, this was the first year of this research, so we're hoping to, you know, redo it again and just, you know, get more data to kind of back up our points. And some of the research that we'll be doing this upcoming year, like I said, we're going to continue to try to pinpoint some of the predators and parasitoids, pathogens that might be controlling these populations. There's likely other animals as well that are getting involved. I'm sure there's plenty of bird species that would be feeding on something like a zigzag larva. Even though they're such a small insect, you know, there's been plenty of times, like I said, where we've gone to places and just haven't been able to find a specimen to back up the feeding damage that we're seeing. We're going to continue to send specimens to Canada, who's doing some genetic research to kind of pinpoint where a lot of the individuals are coming from, whether they're coming from China or Europe, because oftentimes they do come from nursery stock. Like I said, with the overwintering pupa, they've been finding them in the soil in the nursery stock. So we're going to try some emergence traps as well in some of the infested forests that we have to just see how many insects come out of the ground underneath some of our infested trees there. And just the general continuing of mapping and damage severity tracking as well. So for reporting, if anybody sees any suspicious feeding pattern that looks like EZS, you can send photos to our the Forest Health email account, which goes straight to the Forest Health Diagnostic Lab. You can make a report on IMAP invasive, imapinvasives.org, or you can just contact myself if anybody has anything to follow up with or any more specific questions. Feel free to contact me there. And that's all I have.
Thank you, Liam. That was great. It's that's a lot of information about a species that I don't know too much about, so I really appreciate that.
Yeah, absolutely.
Do you see, so what time of year is the defoliation usually?
So you'll see the defoliation damage until the fall, right? Until they drop the leaves. But generally we've seen the worst in like late spring throughout the summer into the early fall when they start to get into that overwintering stage.
And do the elms leaf out again if there's an early defoliation?
Yeah, I mean, generally we haven't seen it get to the point where an entire tree is defoliated, where it needs to reflush those leaves. In areas where they have had that severe defoliation, they have seen it. It's kind of just like a spongy moth, where you'll have like a severe defoliation, and then the population kind of like dampers down as they go through their life cycle. And the tree reflushes, but it's just one of those things where if it's a year after year of this kind of defoliation, then it's not going to take too long for that tree to just up and quit, really.
Poor elm.
Yeah, for real. They've been having a hard time lately.
Yeah. Does anyone else have any questions? Just like before, you could unmute yourself, you could raise your hand, or you can enter in the chat.
I've got a quick one if you don't mind.
Yeah, go, Jeff.
Thanks, Liam. Really appreciate it. Does it have a preference for any particular elm, or is it pretty much an equal opportunity elm pest?
Yeah, so it seems to, anecdotally, it seems, because we're still trying to get into this research, but it seems to favor American and Siberian elm over others. They have been doing some of this research in Canada, I believe, as well, where they found some of like the pretty niche ornamental elm to not, to be unaffected. But across the board, it does seem that they will feed on just about any tree in that genus. Just the most damage with elm and Siberian, really, or American, excuse me.
Other questions? Do they think that it came to Canada from nursery stock? I know that you had mentioned it was found in nursery stock, or it sounds like it was found in nursery stock in some areas.
Yeah, that does seem to be the main theory of how it's been traveling so quickly, I guess, really. I mean, they are great flyers, right? With flying 56 miles in a year.
Right.
But they have found it plenty of times emerging from the nursery stock soil. So that does seem to be the main mode of transportation.
That's disappointing.
Yeah, definitely.
Anyone else have any questions? Well, if anyone thinks of anything, you're welcome to enter it into the chat. Are you staying around, Liam, or you got to get going too?
I do have to get going, but
okay.
I can throw my email in the chat here just in case
Sure.
anybody has anything to follow up.
Yeah, that'd be great. Thank you. I appreciate it. And thanks again for giving the presentation. Like I said, I have a lot to learn about elm zigzag sawfly. It's a species that's, from your maps, it's really exploded
Yeah,
last
yeah,
year,
right.
just last year.
Yeah, right. It just takes a little bit of outreach, and then all of a sudden it's everywhere.
Yeah, exactly.
know.
And it's a good example of like what we see with a lot of invasive species is that we think it's not here, then you start to do some outreach, then all of a sudden it's in a lot more places than you think.
Yeah, I mean, especially with the damage being so low, it is one of those things that you kind of really have to look for, unless it's like one of those Niskayuna situations where they just seem to really thrive in that area. If you have a bunch of elm close together, and then all of a sudden you have a bunch of completely defoliated elm. But
Mhm.
in terms of New York State, it's only been that one situation where we've seen damage that severe.
Mhm. But seems like we can expect more
I
as
would think so.
it's
Yeah. Yeah.
establishing itself. Yeah. Well, thanks again, Liam. That was a great presentation, and I echo what Liam said. Please report if you see it. There's a lot we are still learning about this species and beach leaf disease. Report both of those species when you see them in CRISP. Thank you, Liam, for
Absolutely.
your time and your great presentation. I
Yeah.
appreciate
Thank
it.
you guys. Take care.
Yep, take care. So next on the agenda, I think is I'm going to give some crisp updates. We're finishing the 2023 annual report. Um, some of the impressive metrics that we have in there include that during 2023, the CRISP aquatic and terrestrial strike teams surveyed a total of 6,229 acres of water bodies and uplands and managed a total of 52.7 acres. The area surveyed was an increase of 60% over 2022. While the area managed was down from 2022, but some of the decline in management is due to success of the management over time and a decrease in the area of managed species. In 2023, CRISP led a total of 46 events for 1,135 participants. 23 trainings were offered for identification, reporting, early detection, control, and prevention of invasive species. Other programs raised awareness about spotted lanternfly, beach leaf disease that we just heard about from Jess a few minutes ago, jumping worms, and other emerging species. Programming was offered both remotely, like this, and in person. The most popular program we had was Sarah did a program about aquatic invasive species at the Burke School with 108 participants. Alexa is creating a volunteer database, and we're improving our registration and tracking of volunteers, so look forward to that this year. And we have a lot of programs that are coming up. We have them on the CRISP events calendar on the CRISP website, so please take a look. It's catskillinvasives.com events to see what we have going on. Maybe I should reshare my slides if I can get to that. I just want to reshare the intro slide here. Again. So next on the agenda, we wanted to announce the award for the request for proposals that we put out for 2024. We awarded it to the Otsego County Conservation Association. They'll be continuing the early detection and rapid response program that they started last year as a pilot program. And maybe if Jeff O'Hanley is still there. Are you still on, Jeff?
Yeah, I'm still here, John.
Okay, great. Do you want to say anything about the project?
Oh, apparently I have it. Oh, there we go.
Oh.
Yeah, we're really grateful to have received the funding from you, and we're really looking forward to getting going. For those who are not familiar, the project is aimed at a lot of rapid response to some high priority AIS in the county and immediate surroundings. So we'll be getting some people out on a regular schedule to pull things like yellow floating heart and frogbit, water chestnut, etc. And we're really looking forward to it.
Thanks, Jeff. Yeah, we're really
Yeah, thank
looking
you.
forward to your project, and hopefully we get a lot of volunteers out there, and we're surveying and managing a lot of invasives.
Yep.
So next on the agenda, just wanted to bring up about the CRISP cooperation agreement. It's available for any organization to sign. We've had a number of our steering committee sign it, including the Catskill Center, the Cornell Cooperative Extension of Columbia and Greene Counties, Delaware County Soil and Water Conservation District, Mountaintop Arboretum, New York City Department of Environmental Protection, SUNY Oneonta. And those organizations all signed it last year, and then we've had a flush of some organizations signing it this year. Those would include Adirondack Research, Flying Trillium Gardens and Preserves, Lake Association of White Lake, and the Otsego County Conservation Association, who we just heard from Jeff. In addition, we have other agreements with DEC and Department of Agriculture and Markets. So they haven't signed our cooperation agreement, but we're continuing to work with them to support the mission of CRISP in performing invasive species management. And there are other benefits to signing the proposal for anyone that's interested. You get points on your RFP proposals when we score them. If you put in a proposal for next year's RFP, and you could be considered to be nominated for the CRISP steering committee, so you'd get to pull the levers of power in the CRISP steering committee, help to work on the strategies that we're continuing to work on through that steering committee. I don't know if anyone, maybe Meredith is still there, if you want to add anything to that about the signing the agreement. I'm not sure if Meredith's still on or not. But if anybody has any questions about the cooperation agreement, please send me an email, give me a call. We could talk about it. I'd be happy to talk to you about it. Oh, and Sarah reminded me, just wanted to bring it up to anyone that this may be happening to. We've been observing with the CRISP listserv, which is how we advertise for this meeting and our events that we have coming up, that it's going into spam folders. So if that's happening, please let me know. I'd like to keep track of that. And obviously, if we're sending out things on the listserv and people aren't seeing them, that doesn't help in our advertisement and our getting partners to know what's going on about invasive species management, about CRISP and the CRISP region. So let me know if you've observed that. I'd be really interested in any other reports of the listserv emails going into your spam folders. And next, we have some time on the agenda for partner updates and announcements. I sent out an email the other day just asking if anybody had any announcements they'd like to make. I haven't heard back from anybody as of this morning. But does anyone have any announcement, any event they have coming up, or anything they'd like to mention about their organization? We have a few minutes for that now. Douglas Knopf, you go ahead, Douglas.
Thanks, John. This is Douglas Knopf. I'm the project manager from the IMAP Invasives teams, the New York Natural Heritage Program. I just want to give a couple of quick updates about some things that we have going on right now. So our Hemlock Woolly Adelgid Mapping Challenge is still going on, going to be continuing through next Friday. If anybody has any last minute HWA data points to get in, you can do so. I'll put the link in chat to link to that as well. We also have one update on our species tier. So the IMAP Invasives teams has been working on running the analysis on the invasive species tier data, and we are aiming to have our new data tiers to send to our PRISM partners before the end of this month. So I'll be reaching out to the PRISMs, like I said, hopefully sometime before the end of March with that new data. And that's it for me. Thanks.
Great, thanks, Douglas. Yeah, no, I'll put in a plug again for IMAP Invasives. Please report observations of these species that we've been talking about today, and any observations that you have of invasive species in the crisp region. That that database is incredibly helpful for us in the work that we do, and that's how we plan our management for invasive species. Anyone else have announcement that they'd like to make? I'm not hearing anything. So our next thing on the agenda is to wrap up the meeting. What I'll mention again now, I think I mentioned at the beginning of the meeting, we're expecting our next partners meeting will be in May. We try to do them quarterly. I know that there's some new people on here this time. And the way that we've been doing it the last few years is we've been holding in-person meetings during the warmer part of the year. Usually it falls in May, and then we'll have another meeting that will fall in August, September. And those will be in person. So we're looking for a place in the crisp region to hold a meeting in May. So if you have any suggestions, you could enter them in the chat if you got a chance today in the next few minutes, or you could email me or give me a call. I'd love to chat with you about it. We've, in the past, when we've done more in-person meetings, we tried to move them around the crisp region to give people better opportunities to attend our meetings. And then in the colder parts of the year, November, December, and February, March, we hold a meeting during each of those times. Those are usually virtual. That hopefully is helpful to people to attend. When we have bad winter weather, we can avoid that by having a virtual meeting. So that's been our plan for the last couple of years, and I think that that's the way we'll do it going forward. Our next meeting, we could potentially have, like last year we had a meeting at DEP in May in their Arkville office. We had a meeting at the Catskill Visitor Center in Mount Tremper in September. So those might be places where we could hold future meetings. With that, I'd like, unless anyone else has anything to add at this point in time, I think we can wrap this meeting up, and I appreciate everyone's time. I hope that you were able to learn a lot from the presentations that we had. We were really fortunate to be able to have those two great presentations to learn more about these species that are really, as you can see from the maps, exploding in New York State, and we expect we'll have more impacts over time. So it's good that we can recognize those species and continue to learn about them over time. And I'd like to thank, even though I think they're both dropped out, Jessica and Liam for their time and doing those presentations. Jessica was able to jump in last minute to do that presentation for Kelsey. So as I mentioned, we'll probably have our next meeting in person in late May. We'll let people know as that meeting gets planned and put on the calendar. But thanks again for all your time today. Really appreciate it. Hope that we can be in touch through the season as everybody's getting into the field season this year. Really appreciate your time today. Thank you.